All right, let's dive in. Today, we're going to be digging into a topic that is, and I mean this literally, reshaping the future of our food: plant chromosome engineering. We're going to explore how scientists are moving way beyond traditional breeding to actually design the crops of tomorrow, right now. So, let's just start with the biggest question there is, the one that keeps scientists and farmers up at night. Our global population is rocketing towards 10 billion people by 2050. So, how in the world are we going to feed everyone? That single massive question, it's the engine driving all the incredible innovation we're about to talk about. Because the reality is, we're facing what you might call a perfect storm. It's not just about more mouths to feed. We're also dealing with the intense pressures of a changing climate. And, maybe surprisingly, decades of selective breeding have actually narrowed the genetic pool of our most important crops. So the bottom line is this: the traditional methods, the ones we've relied on for centuries, they just aren't fast enough or powerful enough to keep up with all these challenges. So here's our game plan for this explainer. We'll start with what we're calling the breeder's dilemma. Then we'll define exactly what chromosome engineering is. We'll trace its history from, clunky X-rays all the way to CRISPR. We'll see it in action, and finally we'll look at what this all means for the future harvest. First up, the breeder's dilemma. Look, for centuries we've improved our crops through classical breeding, but we've basically hit a wall. Let's dig into why that is. I think this slide just lays it out perfectly. On the one hand, you have classical breeding. It's like a long, slow game of chance. You're basically just shuffling the genetic deck over and over again and hoping, really hoping you get a good hand. But with chromosome engineering, well, it's a rocket ship. It's fast, it's precise, and it lets scientists choose the exact genetic cards they want to build a winning hand, creating traits that you would literally never find in nature. So that brings us right to the core of it. What is chromosome engineering anyway? Let's get into the fundamentals. Okay, so before we can talk about editing, we gotta know what we're editing, right? So think of it like a massive library. All the DNA in a cell is like all the books in that library. A chromosome, that's like a single, tightly bound volume, a really compact package that holds all these different chapters of instructions, which we call genes. So if chromosomes are the volumes in our library, then chromosome engineering is like being the master librarian with superpowers. You're not just correcting a typo in a single sentence. That's kind of what gene editing is. No, you are rewriting entire chapters, swapping sections between completely different volumes, or even creating a whole new volume from scratch to add powerful new traits like disease resistance right into the plant's master blueprint. Now, this kind of power definitely didn't just appear overnight. So let's take a quick look at the history here and see how this revolutionary science actually evolved. Yeah, the path to precision has been a really long one. It started way back in 1956 with what you could only call the genetic shotgun approach. I mean, literally blasting wheat chromosomes with X-rays and just seeing what might happen. Then in the 60s, chemicals offered a bit more control, but it was still basically a game of chance. The real revolution, the one that changed everything, came in the 2010s with CRISPR, a tool that finally gave scientists the incredible precision they've been dreaming of. And this quote just says it all, doesn't it? It perfectly captures the incredible leap in technology. We've gone from a genetic sledgehammer just causing all this unpredictable damage across the entire blueprint to molecular scalpel, CRISPR, that allows scientists to make edits with surgical pinpoint precision. Okay, now for the really exciting part. Let's look at how these molecular scalpels are being used right now, today, to build the crops of the future. So, one of the biggest and most immediate breakthroughs is just a massive time saver for breeders. Think about it. Creating a new, stable plant variety can take a decade, sometimes more, of patient crossbreeding. Well, a technique called haploid induction can shrink that timeline from years down to a single season. It's a total game changer. So how does this magic trick actually work? Let's just follow the diagram here. Scientists basically sabotage one of the parent plants. They modify a key protein that kind of holds the chromosomes together. You can see that over on the left. So when they cross this modified plant with a normal one, the sabotaged chromosomes are just too weak to stick around. They get completely eliminated. So what are you left with? You see it on the right. A haploid plant with only one set of chromosomes from the normal parent. This creates a genetically pure line instantly, which is just an incredible accelerator for any breeding program. Okay, this next one is really, really cool. It's called traits stacking. So imagine a plant's DNA as like a super crowded bookshelf. Instead of trying to squeeze new books in one by one, what if you could just bring in a whole new, perfectly organized shelf? Well, that's the big idea behind mini chromosomes. They're these tiny custom-built platforms designed to carry a whole stack of new traits. And this slide shows one of the clever ways they actually build these things. Using what they call a top-down approach, scientists use their genetic scissors to essentially chop off the ends of a natural, full-sized chromosome. They strip it down to just the bare essentials, creating this tiny, functional mini chromosome that's basically ready to be loaded up with a cargo of useful new genes. And their potential is enormous. As the research points out, these mini chromosomes could become the go-to vectors, which is just a fancy term for a delivery vehicle, in all of future plant biotech. The cargo ship analogy here is just perfect. They can be loaded with entire suites of traits, like say drought tolerance and pest resistance, and deliver them all in one single go. CRISPR's precision also allows for some, well, major renovations. You know how sometimes a really great gene for, let's say, fantastic flavor is physically stuck right next to a bad gene for low yield? They're linked. Well, as this diagram shows, CRISPR can be used to make these incredibly precise cuts, shuffle entire segments of chromosomes around, and effectively break that unwanted link. This lets scientists keep the good traits while editing out the bad. So what does all of this add up to? Let's just zoom out for a second and look at the real promise of this technology for our future harvest. You know, the promise here is nothing short of revolutionary. We're talking about crops that can defend themselves from pests, that can thrive in a drought, that can produce more food on less land, and can even be more nutritious. So ultimately, this isn't just some fascinating science experiment. It's one of our most powerful tools in the mission to achieve lasting global food security. And that really leaves us with a final, pretty powerful question. For centuries, we've been bound by the limits of natural selection and really slow breeding. But now we have the ability to design our crops from the blueprint up. So what kind of resilient, sustainable, and abundant food system will we choose to build? The possibilities, well, they're really just beginning to grow.